In this tutorial, I'm going to tell you about gene set enrichment analysis, or GSEA. I'm going to tell you how it works, how to interpret the outputs, and how to run it on your own computer. I'm going to cover these topics, and I'll put the timestamps in the description below so you can jump to the section that you're interested in. GSEA is a knowledge-based approach for interpreting genome-wide expression profiles. You can use it to interpret changes in gene expression from, say, an RNA-seq experiment, a microarray experiment. It was developed by scientists at the Broad Institute, and the main reference is a paper here from 2005. So what does GSEA do? It studies sets of genes and looks to see whether they are enriched in your experimental dataset when compared to your control dataset. So it's different from gene ontology and other methods like that because it doesn't just consider genes which are significantly changed. Because when you look at only genes which have significant changes, often you don't have a lot of overlap between related experiments. So in this particular example, I've treated cells with different drugs, and it doesn't look like there are many genes in the middle which are changed by all 4 of the drugs. However, phenotypically, the cells respond very similarly to these drugs. So simply looking at significantly changed genes and comparing between experiments or between datasets doesn't always give you a good impression of what's actually happening in the cells. What GSEA does is it looks at all of the genes, not just those that are significantly changed, and it looks at even the smallest changes in gene expression to see whether there are changes across a pathway or across a set of genes related to a particular phenotype. And the logic behind this is that if you look at significantly changed genes, there might be one in a pathway which is upregulated by 20-fold, but just having one gene change in a pathway might not do a whole lot. However, if most genes in a pathway are upregulated by 20%, then that could give a much higher flux through that pathway or indicate a big change in phenotype in the cell. So by looking at all the genes in a pathway and small changes in gene expression, you might see much more meaningful changes in the cells. So this is the first figure from that paper from 2005, and it illustrates the method of how it works. So here we have your data set. So A would be your experimental and B would be your control. And you have essentially data, quantitative data from your experimental and control. So these could be expression, it could be count data, it could be t-tests, so something quantitative. And what the software does is it ranks each gene. So it will take an average across all the samples in class A for each gene, and then rank those genes based on the counts that you have provided. And then once it's ranked that data set, it then takes a gene set which you have chosen. So there are gene sets within the software. You can create your gene set. So for example, this could be a set of genes that are all linked to cell cycle control, for example, or a set of genes that are related to MAP kinase signaling. And the software looks at where each of these genes matches up with your ranked list of genes here. And so just by looking at it, you can see that more of these genes, so each line here is a gene in that set. So there's more of these lines up near the top of the data set and fewer of the lines near the bottom of the data set. So you can immediately get an impression that these genes are enriched in genes that are more highly ranked in this data set. But how do we put a number on that? So what this software essentially does is it quantifies this enrichment of these genes near the top of this data set. So the way that it works is it starts at the top of the list and moves down gene by gene. And it creates an enrichment score, which is called ES. The S in brackets refers to gene set S. If the software encounters a gene that is also present in the gene set, it will increase the enrichment score. And it increases the enrichment score by a value related to that counts data, so the size of that gene expression that you have here. So for each gene that it encounters that is also found in the gene set, it increases the enrichment score like this. For each gene it encounters that is not present in the enrichment set, it decreases the enrichment score. Not by the same amount, not by that correlation, but by an amount that's proportional to the size of your data set. And so what you tend to get if your gene set is enriched in your expression set is a graph that looks a bit like this, where the enrichment score climbs up as it encounters genes that are in the gene set, and then as it encounters fewer genes that are in the gene set, the general trend is downwards like this. And so the shape of this curve here relates to the arrangement of the genes in the gene set compared to your whole expression set. It then calculates some numbers. So the height of this peak here is the enrichment score. And so the size of this enrichment score gives you a number for how enriched the gene set is in your data set. And it also gives you a probability for how likely this enrichment score is to be significantly different from random. These are the sorts of plots that you might see in a paper that has used GSEA to analyse changes in gene expression. Along the x-axis we have the ranked gene set ordered from left to right. So on the left are genes which had the highest expression in the experimental compared to the control, and that decreases. So over here we have genes with the lowest expression, and the colour in this bar represents that as well. These bars represent the different genes in the gene set that was being tested. So you can see here we have lots of bars on the left. So these are genes which are enriched. These are highly expressed in the experimental sample compared to the control sample. And over here we have another cluster of genes from that gene set. So these are genes that are more expressed in the control sample or are decreased in your experimental sample. And here is the graph of the enrichment score. So you can see it increasing during this point where there are lots of genes, and then where there's no genes, that score decreases. And then at the end, it's actually dropping below 0 because these genes are more highly expressed in the control. And as I said, you might get an enrichment score. So here it's 0.4 and a p-value here 0.0198 to suggest that this particular gene set is enriched in the experimental sample. So in this example, these are genes involved in the G2M checkpoint. So we can see that the experimental had an enrichment of those genes. Conversely, from the same data set, now we're looking at genes involved in rRNA metabolic processes. And we can see if we look at the 0 of the enrichment plot, now most of the genes in this particular gene set are actually enriched over here on the right. And this is telling us that these genes are downregulated in the experimental sample and have higher expression in the control. So the important thing to look at is where is the 0, and does the graph go above the 0 at the start, or does it go below the 0 at the bottom? And so here the enrichment score is minus 0.558. So these genes are enriched in the control rather than the experimental. So how do I perform this? The software that you use you can download from the GSEA website after registration, and they have an excellent user guide that helps you understand the details of what's going on. So this is the GSEA website. So you can click on links to download the software. You can look at the molecular signatures database, which is where you can download gene sets, and I'll talk about that later. You can also view documentation. And the most useful thing about this is the user guide. So here's a link to the user guide. It's also in the description below. And this can give you details about your file formats and how to use the software. So the input files that you need to create are the expression dataset file and the phenotype labels file. The expression dataset file looks like this. You can create it in Excel, save it as a tab delimited text file with the extension gct or txt. So it is arranged with your samples in columns and the genes in rows. The very top left cell must contain this text, hashtag 1.2, and don't change that. In the next row on the left is the number of genes you have in your file. So here, typically you might have 50,000-60,000 if you've got an RNA-seq experiment. That number will be different if you're using microarray data. The next cell on this row tells the software how many data sets you have or how many columns. So in this case I had 12 columns, so here this would be the first one, 2, and then the rest of the 12 I'm not showing. The next row contains the headers for the columns. So the first header is name, don't change this, capital letters, and this is telling the software that this column contains the gene names. The next column contains the word description, and then just put NA for all your genes. Don't change this, just put NA. The next headings will be your sample names. So don't use any spaces or unusual characters, just use numbers and letters. And each of these sample names must be unique. Then for each sample, for each gene, you have your expression data. If these are RNA-seq data, they must be normalized in order to compare different samples. So for example, if you've been using DSeq2 software, use the counts output from that. The second file you'll need to prepare is the phenotype labels file. Again, you can prepare it in Excel and save it as a tab delimited text file, but this time the extension should be .cls. So this file only has 3 rows. The first cell should contain 3 numbers. The first number is the number of samples that you had in that expression dataset that we were just looking at. So if you remember, I had 12 samples, so I have 12. Then there's a space, and then there's the number of groups. So my 12 samples I put into 2 groups. Then you have a space, and then you have the number 1. So number 1 will be the same whatever file you are preparing. In the second row, you need a hashtag, then a space, then the name of the first group, then a space, then the name of the second group. And then the third row essentially tells the software which of your samples go into which group. So this needs to be the same order as the samples on your previous expression dataset file. And so the software is going to put the first sample into whatever group you've listed here. So it's going to put my first sample into the EBZ group, second, third, fourth, fifth, sixth samples are all going to go into that EPZ group. And then the final 6 samples are all going to go into the control group. So the order of these cells is important, and it relates to the order of the samples in the expression dataset. Now here I've just got 2 phenotype groups. You can have as many phenotype groups as you want. However, GSEA will only compare 2 groups at a time. Now this is using categorical phenotype groups. Samples are either in one category or in another. You can also compare samples in a time course, and if you wish to do that, look at the user guide to understand how to use continuous phenotype labels. So a key part of GSEA is those gene set files to compare with your expression dataset. So what ones are available? So there are several collections of gene sets available within the software. You can also search and download individual gene sets from those collections that are more focused on your particular question or hypothesis. And you can also create your own gene set from other sources that might be more specific or more focused on what you want to know. So it's really important to understand a bit about the gene sets within the software before you just go ahead and randomly use them. So within the molecular signatures database on the GSEA website, you can learn more about them. So the collections that are available are listed here, and there's 8 major collections. And the hallmark gene sets is perhaps the most useful one to start with. So if we click on collections here, we can find out a bit more about them. So the hallmark gene sets represent specific well-defined biological states or processes, and they were generated by studying a large number of different gene sets and resolving them, so there's minimal overlaps in genes between the different gene sets. If we click here, we can look at the different gene sets that are available, so you can see them here, and you can click on them to get an idea of what's in there. So in the hallmark adipogenesis gene set, we can see that this contains genes upregulated during adipocyte differentiation. If we look at some of the gene sets related to signalling, for example, let's look at this one, TGF-beta signalling, we'll see that this contains genes that are upregulated in response to TGF-beta signalling. So it's not genes that are part of the TGF-beta signalling pathway, it's genes that are upregulated in response to it. If we look at some of the other gene sets that are available, so we have positional gene sets corresponding to different chromosomes. Within the C2 curated gene sets, there are several different collections, including several to do with pathways. So we have gene sets to do with from the KEGG database, and we also have gene sets from the Reactome database. So if we click on that to see what Reactome has, so here we have loads, 1500 different Reactome pathways. Other gene sets, we have regulatory target gene sets, so we have got things to do with microRNA or transcription factor binding. Down here we have some computational gene sets, ones related to cancer. And then down here we also have gene sets related to gene ontology. So we have biological processes, gene sets, cellular component and molecular functions. And you can see within the biological processes collection, we've got over 7,500 gene sets, which is a lot to be working with, but you can look at them all here. C6 contains oncogenic signatures, and these are generally derived from experiments other people have done. So for example, let's look at P10. So P10DM, that will mean genes downregulated upon knockdown of P10. So click the link, see what's involved, and you can see where it was generated. So there's lots of gene sets from different experiments related to oncology. So you can see that some of those collections contain more than 1,000 different gene sets. And that's great for getting an overview of what's happening. But when you start to look at the false discovery rate, the Q value, for every additional gene set that you're testing, you're going to reduce the significance of those that are pulled up as being changed. So you might want to focus your gene sets on a particular question or biological system related to the question that you're addressing. And so you can use the search function to pull out gene sets from all those databases that are related to a particular pathway you are interested in. And as I said before, if you want to create your own gene set from other data that you have, there's easy instructions within the GSEA website about how to make that file, and that's not very complex. How do I use GSEA software? So this is what the software looks like when you open it up. So the first thing you need to do is load your data up here at the left. So we click on that, and we need to browse for files. You can then click on both of your data files, so the gct file, and if it loads up correctly, it'll give you this little message that says there were no errors. And then you can go and upload the second one. Here we have my CLS file. And again, it tells me there are no errors. And it lists them down here, and you can find them again if you need them later to rerun. So that's our data loaded. So now we click on the Run GSEA box here. So expression data set, so this is where you now you've loaded your data, you can choose it. So this is your GCT file with all your count data in it. Gene sets database, so this is where you choose which gene sets you want to use. So by clicking on this little 3 dots at the right, here we have all those collections of gene sets that we were looking at on the website earlier. If you have downloaded an individual gene set, you'll find it here, but we're just going to look at these collections here. So when you're first starting out, probably the first ones to look at is the hallmarks. It's going to give you an unbiased overview of many pathways and cell states. So we're just going to click on that, and I guess just to say, so as you go down this list, so this is version 7.1, as you scroll down you get to the older version 7.0, 6.2 if for some reason you want to go back to an older version. So we'll use the most recent and click OK. Number of permutations, so keep that at 1000. This is the number of times that it tests things. So phenotype labels refers to your samples that you want to compare with each other. So if you click on these 3 little dots here at the right, and it is basing this on your CLS source file. So in this particular example, in that source file, I have 15 samples that are put into 5 classes or 5 groups. And what the software is asking me to do is say, well, within those 5 groups, which 2 do you want to compare to each other? And quite usefully, it will let you choose one of them compared to all the others. So where it says rest here, it's saying, well, this is one sample and it's going to combine all the others into a group. And so you can compare one group with all the rest of them. Or you can, if we scroll down, we can add individual groups, this group compared with that group, or my control group compared with this one. So you choose which comparison you want to do. So let me choose this one here, EPZ versus control, and you just click OK. Collapse, that's fine. Permutation type, so this is to do with how it calculates the p-value. So it's going to compare your expression data set with the gene set, and then it's going to say, okay, well, if that's the enrichment score we got for that comparison, what happens if we muddle everything up? What would be the random control? And so it can either muddle up your phenotype labels, or it can muddle up your gene labels. So phenotype labels, they only recommend using that when you have at least 7 samples per group. So 7 samples compared with another 7 samples. If you don't have that many, choose the gene set option. That means it's going to be randomizing the genes rather than the phenotype labels. Chip platform might sound a bit odd if you've used RNA-seq data, but it's just the name just derives from using microarrays in the first instance. And essentially what this does is it converts your names, your gene names in your file, into the gene names that the software works with. So again, if you click these 3 little dots, you can choose the one that's most useful for you. So for example, if you use the human AFI array set with this particular number, you click on this and it'll give you the right annotation file. So here I've used ensemble gene IDs from human, so I'll click on that one. You could also download one from the website and use that from your C drive. So once you've chosen that, you click OK. Now we're going to look at the basic fields. This is important to choose these options. So the first option here is where you're going to save your data. And then here you need to give it a name. So it's best if you give each run of GSEA a different name. So I tend to just put in the name of the samples that I'm comparing. So we chose G7 EPZ versus control. And it's important to write down, well, which gene set were you comparing it with? So we chose hallmarks, because otherwise you can end up running lots of different comparisons and you forget which was which. Everything else you can keep as it is. Signal to noise is the default metric for how it's going to rank the genes. These refer to the gene sets, so it's not going to look at any gene sets which have fewer than 15 genes, and it's not going to look at any gene sets with greater than 500 genes in it. Most of the advanced fields you don't need to look at, but there's one worth mentioning. When we come to the output, it is only going to generate graphs for the top 20 gene sets that it pulls out. Now there may be cases where actually you'd really quite like to look at a plot for one of the gene sets that are later down in the list, so you might want to change that number, but everything else you probably don't need to alter. So once you've chosen your options, then you click Run down here at the bottom, and maybe I should just mention this question mark here is online help, and that will take you to a relevant place within the user guide to help you choose which options that you need. And then we'll click on Run and Execute, and down here we now get a little status box, so it says it's running, it's downloading that annotation file for us, and depending how many samples you have and how many gene sets are in that collection, that will affect how long this takes to run, usually between 2 and 10 minutes. When it's finished running, this will change to either fail if something's not worked, or more hopefully success. So you can click on that success. This now gives you a HTML report which it will display in your browser. So what are we looking at here? So here we're saying it's looked at the EPZ samples, and it says how many gene sets appear to be enriched in your experimental sample. So it thinks that 42 out of the 50 it studied are upregulated in that sample. It says 24 gene sets are significant with a false discovery rate of less than 25%. 12 are significantly enriched at nominal p-value less than 1%. And 18 at a nominal p-value of less than 5%. So we can click on Snapshot of Enrichment Results, and that will show you lots of different plots for the different gene sets. In order to see a detailed report of the enrichment results, we should click on this link here. So here each row is a different gene set that it was testing. You can click on the name of the gene set, which takes you to the website to tell you what was in that gene set. Size here means that's the number of genes that were in that gene set. So there were 200 genes related that are downregulated in response to KRAS signaling. ES, that's the enrichment score. So that was the height, maximum height of that peak in that graph. And so the software, and so the ones at the top will normally have the largest enrichment score. NES refers to the normalized enrichment score, and that refers to how many genes were in the gene set. And so it's a better comparison to compare. So this gene set has 200, this has 44. So this is a better ranking metric to compare these two gene sets. This next column is the nominal p-value. So this is just looking at data within the one gene set. How likely is it that this gene set is significantly enriched in your data set? And so here we have ones, as you can see, these are really highly enriched, highly significant, and then down here, down towards the bottom, we have gene sets that are not significantly changed. The next column is the false discovery rate Q value. So this is a correction for the fact that you've been looking at 50 different gene sets. It's a correction for multiple testing. And essentially what it means is that for those gene sets that were significantly enriched, so ones that had a significant nominal p-value, what is the chance of those being a false positive? So this number here, 0.023, says there's a 2.3% chance that this KRAS signaling is a false positive. So typically a cutoff for FDR might be 0.25. So if we look down here, so that might mean everything from number 24 upwards you might consider to be worth looking at. The family-wise error rate p-value is really a statistic which the software used initially in its first iterations, but then the researchers developed their algorithm and their statistics, and so they don't recommend using that anymore, but they keep it in just in case you want to look at it. Rank at max tells you within your list of genes, which gene was it where that enrichment score was highest. So how far down the list of, let's say, 60,000 genes was the peak of that gene set. Then we're looking at the leading edge. And so the leading edge refers to all those genes in the gene set before the peak of the enrichment score. So it tells us that 34% of the genes in the gene set are part of that leading edge. So 34% of the genes in this KRAS signaling down were particularly highly enriched in your list of genes. List equals 14%. That tells us that out of your list of 60,000 genes, 14% of them were in that leading edge, so before the peak of that enrichment score. And signal is some combination of the tags and the list number. So to get a graphical output of this, we click on this link where it says details. So here it's summarising what we've just gone over: the enrichment score, normalised enrichment score, nominal p-value and FDRQ value. And here is your standard enrichment score plot. So you can see that these genes in KRAS signaling down are highly enriched near the start of this gene set. So it goes up very steeply and then decreases as the genes in the gene set are less densely clustered. If we scroll down, here we have a list of all those genes in that gene set ranked by where they occurred. So the very first gene that's listed here, that'll be this one here, and the next one, and so on. So they're ranked by the order they appeared. And so here, rank in gene list, that is the number within your data set where this particular one was found, and that's the running enrichment score. So as it increases, it's going to increase. And then core enrichment, is it one of those leading edge genes? So yes. And as you scroll down, then here are the genes which are not significantly enriched. So if we scroll down further, now we have a heat map, a cluster. And again, these are those genes that we were just looking at. And here are the samples we were comparing, the EPZ versus the control, where red is highly expressed and blue is low expression. And it just shows the top 50 genes. So it's not all the genes that are significantly enriched, it's just the top 50. And this is a really useful sense check to see whether the data that you're getting makes sense and looks clean. So here we can see all these three samples here do look very different from the three control samples, certainly at the top of the list. And as we scroll down, we have some genes that are not changed, and then down at the bottom it's switching the other way, where it's control genes that are higher expressed compared to the experimental. So it's really nice to look at that heat map. And then down at the bottom we have, this is a graph to show how they worked out the p-value, and essentially what this plot shows is if you were randomizing those genes, looking for a null distribution to compare with, this is what it would look like. And because the enrichment score here was, what was it, 0.4, 0.5, 0.58, so 0.58 on this graph is going to be way over here, so you can see the p-value is going to be really low. It's going to fall within a tiny proportion of this curve, so it's not random. And there's two peaks because in this direction this is your positive enrichment score, and in this direction this is negative enrichment score. So if we go back to this page, we can also see the same sort of data for the opposite comparison. So now they're looking at the control samples how many gene sets are significantly upregulated in the control compared to the experimental. So there's many fewer gene sets that seem to be enriched in the control. And again, we can have those same links of all the graphs and then the detailed data. Just to show you what this data will look like on your hard drive. So for each run of the experiment, it saves all the information in a single file. And if you click on it, it looks a little bit daunting because it's showing you all the icons. So if we change that to details, so it essentially gives you all those pictures of all the different GSEA plots. It gives you lots of Excel files and reports and things. But if you want to recreate that HTML page we were just looking at, we can look for either of these. So we've got GSEA report for the EPZ or the GSEA report for the control. So as long as all your files are in the same folder, if you click on one of these HTML GSA report files, you'll open up that same website as you had before, like this. So the next thing I want to tell you about is leading edge analysis and how to run that on your GSEA files. So to perform leading edge analysis, you click on this next box here, and then you'll need to load up data from a GSEA analysis you've just done. So click on the two dots, and here we can upload the hallmarks and load GSEA results. And so this is now the list of those 50 gene sets with the number of genes per set, the enrichment score, nominal p-value, all those things we were talking about. So as I said, leading edge is the genes that are found before the peak of the enrichment score. And so what leading edge analysis does is it says, well, okay, we've got quite a few gene sets here which seem to be significantly enriched. Are there any leading edge genes in common between those gene sets? So let me illustrate that for you. So basically you need to select several gene sets. So I'm going to select all those with a nominal p-value less than 0.1. Okay, so then you can click on Run Leading Edge Analysis. So we have 4 different outputs here. And if I just scroll to the right of this one, each row in this heat map is one of those gene sets we've just looked at. And if you hover over a square, again, it will tell you which gene set it was in. And up along the top, these are all the genes present in the leading edge of each of these gene sets. And so you can look at the overlap. Now we can zoom in a little bit. We've got a file button here to save the data, or we've got some display options. So by increasing the grid size, you can zoom in a bit, and we can also click and drag to make a bit more space for each particular image. So click and drag. Okay, so here we can see here, now this gene is present in 3 of the different gene sets, and we can see that this gene is VCAM1. So by sliding along, you can see whether there are some genes that are present in several of these enriched gene sets. So maybe there's a few genes in there which are really driving a lot of these changes. So here's one that's present in 4 gene sets, for example. Another way of looking at the overlap between the gene sets is this diagram in the top right. So these are the different gene sets along the top and along the side, and essentially we're looking at overlaps between them. And so if there's overlaps between the leading edge genes, you will see them in sort of fainter green boxes. So if this was pale green, it would mean there'd be overlap between this gene set and that gene set. And in this particular case, there's a bit of green here, so that means that there's some overlap between the genes in the estrogen response late and estrogen response early. So that makes sense. And then the other graph we can look at, so these are listed by gene and how many gene sets they appear in. So again, we can change the view on these a little bit, and you can also zoom in slightly by clicking and dragging. And here we can now see which genes they were that we saw right at the left-hand side of this. And Jacquard is another analysis of overlap between the different sorts of gene sets. If you want this data as a HTML report, you can go back to the GSEA results tab up here, and you can click Build HTML Report, and so that gives you an output with links to some of those different leading edge outputs. So the last topic I'm going to cover is how to visualize enriched gene sets as a network. So what this means is that if you have several gene sets that are highly enriched in your expression data set, how can you visualize the commonalities between those? So I said before with the hallmarks gene sets, we had 50 different gene sets, and they've been curated, so there's not an awful lot of overlap between the genes in those different gene sets. However, if you are running Reactome or gene ontology, there will be a lot more interaction. And so here we have an analysis run using the Reactome dataset. And if we look down this column of the nominal p-values, you can see there's loads of gene sets that are really highly significantly enriched. It's quite hard to work out what's going on. So we have something to do with the neuronal system, muscle contraction, complement, G proteins. It's hard to know what's going on. Is there a story here? You've just got a whole list of different pathways. So we can use network analysis to look at the interactions or the overlaps between these different sets. So we do this within that GSEA software and this enrichment map visualization box. So we click on that, and the first thing it says is please launch Cytoscape with the enrichment map plugin before continuing. Cytoscape is a program you can download, and it runs lots of different apps for looking at networks and enrichment and pathways and things, and you can download it easily and run it on your computer. This is what Cytoscape looks like when you've downloaded and installed it. And then what you need to do is load the enrichment map app. So we go to Apps, App Manager, and search for enrichment map. And then you would install it. So I've already installed it. So now that's ready to go. So I can just minimize that. So now we're going to choose which GSEA results I want to analyze. So as I said before, I don't find a lot of interaction between the hallmarks gene sets, but there's a lot going on with the Reactome. So select the file, load the results. So that's the data set there. What you may then need to do is make sure it's looking at the right expression data set. So you can click on these three dots. So this is that data file, the folder from the Reactome. We want to click on this EDB folder, and then you want this long file name which got .rnk at the end. So that's your expression data set. Okay, so now we need to choose which gene sets to include in the enrichment map. And so the default values are p-value of less of 0.005, so only gene sets with a p-value less than 0.005 will be included, and only gene sets with a FDR q-value cutoff less than 0.1 will be included. Now this is really important to think about these cutoffs, because if you set these cutoffs too low, so you only have 2 gene sets, well, you can't really look at a network between 2 gene sets. And so but equally, you don't want to be looking at any old gene sets which are not significantly enriched. So it's important to choose p-value cutoffs and FDR cutoffs that are meaningful, but that give you a network that's worth looking at. So I'm going to choose 0.05 here. And if I click on Advanced Options, you can choose how much overlap between the gene sets there is for it to count as an interaction. So the default is 0.5. So if you increase the cutoff, that means that you're going to make the cutoff more stringent, so you're going to see fewer overlaps between the gene sets. And if you make the cutoff less stringent, you're going to see more overlap. So essentially this is going to look at genes that are found in both gene sets. So there's a line between these two genes if there are genes in common between them. So the default is fine, but you can change that if you want. So we click on Build Enrichment Map. So that tells you it's run it and it's created it in Cytoscape. So we'll go to Cytoscape. Oh, and there we go, that's your enrichment map. A bit hard to look at initially because of all the writing. But what you can do is you can click, these are nodes, you can click on these nodes and drag them so things become a bit clearer. Okay, so now we can start to see that these nodes here, so this one is the gene set for neurotransmitter receptors and postsynaptic signal transmission. This is the gene set for GABA receptor activation. And so all these belong, they have overlaps in the genes in the gene sets. And so red means that they were positively enriched in your expression data set. Blue means that they were negatively, this gene set was negatively enriched. It was found more in your control. And if I drag some of these out here so we can read them, these ones are to do with mRNA, for example. So you can, there's a lot of options within Cytoscape for how to manipulate the image, what you can move, how you can export it, and how you can make this image look useful for you. So just to mention, the size of the node, the size of the circle, refers to the total number of genes in that gene set. Okay, so you can hover your mouse over that node and it tells you there, okay, this is the reactive neuronal system. If you click on it, all the genes in that gene set appear down here with that ranking, and you can scroll down and see, there we go, see the ranking score as it changes. Here it tells us the total number of genes in that gene set. So this neuronal system had 410. This smaller node here has only got 60. So that's the total number of genes. And the line shows that some of the genes in the GABA dataset are also found in the neuronal dataset. So you can see that this is potentially a way of making much more meaning from a long list of gene sets that were enriched by clustering them and grouping them into networks of functional similarity rather than just a long list of titles of pathways. I hope you found this tutorial to be useful. Do us a favour in return and click the like button and leave a comment below to let us know what you liked or what else you would like us to cover in the future. We have lots of other tutorials, lectures and interesting videos, so do browse our channel page and consider subscribing. Thanks for watching.